I want to talk about cell division and mitosis. Cell division has as its goal taking one cell and at the end of a process having two complete identical cells. Mitosis is a part of cell division. Mitosis is taking your DNA, your chromosomes, making copies of those chromosomes, and putting one set of each of those copies of your chromosomes into a new cell. So mitosis is part of cell division, and cell division includes mitosis, but has some other phases that we're going to look at. Let's start with a cell that has only 2 pairs of chromosomes. Humans have 23 pairs of chromosomes, but trying to do all 23 pairs can get rather confusing. So we're going to use an imaginary organism that only has 2 pairs of chromosomes, and the pairing is important. This is why we're going to do colors. Here is the nuclear membrane, the nucleus, that has the green chromosome paired to the blue chromosome, and the red chromosome paired to the purple chromosome. Here I have labeled some of the parts of this cell. This whole structure in here is the nucleus that contains your DNA, and the membrane around it is called the nuclear envelope. And then my chromosomes are in pairs, the first pair and the second pair. Now I want to talk about pairing of chromosomes. Keeping your pairs straight is imperative, and this is why. Each one of these chromosomes is a list of base pairs, guanine to cytosine, thymine to adenine. These pairs make up genes or alleles on various parts of the chromosome. So, for instance, as an example, perhaps hair color has a gene here and a gene here on this pair of chromosomes. Perhaps eye color might be here and here. You can't have a cell at the end in which the blue is paired up with one of these or the green is paired up with one of these, because each pair of chromosomes has to be paired up with a complementary chromosome that has the same gene loci or gene locations, even though there might be different genes. Perhaps you carry a gene for brown eyes and blue eyes, and perhaps you carry a gene for brown hair and blonde hair. When you talk about genetics, you'll understand this a little bit better, but it is important to keep these pairs together. At the end of mitosis and cell division, we want to make sure that this is what our cell looks like. So this is where we're going to start, and the nuclear envelope has now been dissipated, and then we're going to end up there. Now, to help us keep track of these chromosomes through this process, I've labeled them pair 1 and pair 2. And pair 1 consists of chromosome 1 and 2, and pair 2 consists of chromosome 3 and 4. So I will refer to these numbers as we go through our process. Here is our cell in prophase. By the time nuclear division gets, or mitosis, gets to prophase, you have 2 identical copies of chromosome 1 connected by a centromere, 2 copies of chromosome 2 connected by a centromere, 2 copies of chromosome 3 connected by a centromere, and 2 copies of chromosome 4 connected by a centromere. But notice the pair 1 still has blue and green, and pair 2 still has red and purple. The next stage is going to be lining these things up. Here we are in metaphase. In metaphase, the pairs of identical chromosomes, we have 2 copies of chromosome 1, 2, 3, and 4. They're all connected with a centromere, and when they're connected with a centromere, remember, we call them chromatids or sister chromatids. They all line up along the diameter of the cell, right down the middle. Now, we will also see some other structures appear here to help us facilitate separation of this nuclear material. Here we are still in metaphase, but I have added the centrioles at opposite poles of this cell. We still have our pairs of sister chromatids right down the center or the diameter of this cell. And these centrioles are going to play a very important role in helping to pull these sister chromatids apart. Now, centrioles come from centrosomes. They're very complex structures. We could run a whole video on those, but I'm going to try to keep it simple here. Let's go forward. Here I've added the spindle fibers, which connect to one copy of each sister chromatid, and the other end connects to a centriole. The centriole and these spindle fibers are what are going to allow us to pull apart these pairs of sister chromatids. Remember, when they're joined together by a centromere, they are sister chromatids. We're now in anaphase. The pairs of sister chromatids have been pulled apart by these spindle fibers attached to the centrioles. So now each of these are individual chromosomes. Once again, here's chromosome 1, 2, 3, and 4. This would be pair 1. This would be pair 2. Here we're in telophase, and telophase is determined, one of the best characteristics that determines telophase is this cleavage furrow. This is going to lead to cytokinesis. We're going to actually split the rest of the cell. Notice we have one copy of each chromosome on each side. And when we split the cell through here, we'll then have two identical copies. I have went ahead and added this little contractile ring of fibers. This little group of fibers work just like a drawstring, and they squeeze the cell into two new cells. Notice on either side of what will become the division between two new cells, we've got a complete set of chromosomes. There's pair 1, there's pair 2, pair 1 and pair 2. Here we have finished cytokinesis, basically squeezing that one cell into two, and we can see that the chromosomes are starting to orient and move towards their proper pair. This will be chromosome 1 and 2. They form pair 1. This will be chromosome 3 and 4. It will form pair 2. Here the pairs have lined up a little more formally. Pair 1 with chromosome 1 and 2. Pair 2 with chromosome 3 and 4. Notice that this is the same cell that we started with, but now we have two copies, two identical copies. The next stage, we're going to show the nuclear envelope reforming. Here we have reformed our nuclear envelope around the nucleus. The nucleus contains two pairs of chromosomes, pair 1 and 2. Pair 1 has green and blue chromosome, or chromosome 1 and 2. Pair 2 has a red and purple chromosome, or chromosome 3 and 4. It is very important that you match the proper chromosome up in each pair in order to be correct when you're drawing or explaining phases of mitosis. This is often why we use the colors. Hopefully this discussion has clarified the various stages of mitosis and cell division.